Hi guys, welcome to chapter 16. We're going to talk about the molecular basis of inheritance. Okay, to get started, let's review an overview. This is life's operating instructions. Alright, in 1953, James Watson and Francis Crick shook the world. They just discovered, whatever word you want to use there, an elegant model of a double helix to show the structure of deoxyribonucleic acid, or DNA. DNA, the substance of inheritance, is the most celebrated molecule of our time. Heredity information is encoded in the chemical language of DNA and is reproduced in all the cells in your body. Alright, it is the DNA program that directs the development of many different types of traits. Alright, 16.1. The topic of discussion for 16.1 is DNA, the genetic material. Okay? So to give you a little backstory, a little history here, early in the 20th century, the identification of the molecules of inheritance loomed as a major challenge to biologists. Alright? The role of DNA in heredity was first worked out by studying bacteria and the viruses that infect them. Alright? Frederick Griffith was a researcher that was studying Streptococcus pneumoniae, a bacterium that causes pneumonia in mammals. He worked with two strains of the bacterium, a pathogenic and a non-pathogenic strain. His test subject was a mouse, alright? So he found that when he mixed heat killed, which means raising the temperature of the solution that the bacteria is in to a temperature that is damaging to that cell's DNA, so heat killing that bacteria, he found that when he mixed those heat-killed remains of the pathogenic strain with living cells of the non-pathogenic strain, some of these living cells became pathogenic. They were able to grow the capsule that protected the pathogenic strain, so those cells were able to become pathogenic. Okay? What this figure here is showing are your first two controls, a control of living, they call them smooth cells, B cells, okay, and a control of living R, or rough cells, okay. Smooth B cells are pathogenic, rough cells, non-pathogenic. B cells, the mouse dies. Living R cells, the mouse lives, because they're non-pathogenic. If the mouse was given heat-treated or heat-killed B pathogenic cells, the mouse stayed healthy because it had been heat-killed and that cell was no longer viable. Then, their last test, they mixed heat-killed pathogenic cells and living R cells, and they found that the living, as a result of blood test after this had been injected and had a chance to affect the mouse, they found that they ended up with living B cells, or living pathogenic cells, in the blood of the sample. So that means that DNA within that heat-killed B cell was still able to influence the living R cells, even though that cell was no longer, even though the pathogenic cell was no longer living. So they concluded that there some form of transformation had taken place. Okay? Griffith called the phenomenon transformation, now defined as a change in the genotype and phenotype due to an assimilation of external DNA by a cell. So that's where it takes on the characteristics of external DNA. Okay. Evidence that viral DNA can program cells. Additional evidence for DNA as the genetic material came from studies on a virus that infects bacteria. Alright, viruses that infect bacteria are called bacteriophages. They are widely used as tools by researchers in molecular genetics. Alright, bacteriophages make pretty decent markers to test for different things. Okay. Our researchers, Alfred Hershey and Martha Chase, performed experiments showing that DNA is the genetic material of a phage known as a T2 phage. Alright, here is the Hershey and Chase experiment. This is a fairly famous experiment. It is discussed often in many different biological science courses. So what the process that they used, they worked through 2 batches. They mixed a radioactive labeled phage, radioactive sulfur is what they used. They mixed a radioactive phage. It had the radioactive sulfur incorporated into the phage protein. That's what this pink is showing here. The radioactivity was incorporated into the phage protein. Alright, that's what's happening here. They agitated in the blender so that they lost these excess empty protein shells after the phages infect the cell. Okay, after they release these empty protein shells, they centrifuge the mixture of the cells, alright, to form a pellet at the bottom that can be analyzed. And then they measure the radioactivity in the pellet and in the liquid. What they found in batch 1 was that the liquid was radioactive where the pellet was not. So, within the cells that they were studying, they were not radioactive. Okay? Batch 2, they mixed radioactive phosphorus with the DNA, and that's the blue. Alright, again with the same, mix it with the bacteriophage, so when the bacteria invades the cell, bacteriophage invades the cell, releases its DNA, they centrifuge it to get rid of the excess empty shells, and then they centrifuge it. What they found at the end of centrifuging here was that the pellet was radioactive, whereas the liquid was not. So, when they tested the individual cells, there was radioactive DNA, phage DNA, in those pellets. So this is how phage proteins are able to take over that bacterial cell's genetic material. It's all through the DNA. They concluded that DNA, not the protein, is the T2 phage's genetic material. Okay? Prior to the 50s, it was already known that DNA is a polymer consisting of nucleotides, each consisting of 3 components. You should remember these. Make sure you know what these 3 components are. Nitrogen base, a sugar, and a phosphate group. Alright. Erwin Chargaff analyzed the base composition for DNA from a number of different organisms. In 1947, Chargaff reported that the DNA composition varies from one species to the next. This evidence of molecular diversity among species made DNA a more credible candidate for genetic material, because there is identifiable genetic diversity. So they evidence this to that molecular diversity. The diversity goes down to the molecular genetic level. Okay? Once biologists were convinced that DNA was the genetic material, the challenge was to determine how the structure could account for its role in inheritance. Alright, so this is where Maurice Wilkins and Rosalind Franklin step in. They used a technique called X-ray crystallography to study molecular structure. Rosalind Franklin herself produced a picture of the DNA molecule using this technique. Okay, as shown right here. Okay. Watson and Crick deduced that DNA was a double helix through their observations of the X-ray crystallographic images of the DNA. So through analyzing this photograph here and other similar photographs that they found from Franklin's studies, they were able to determine that the double helix is the shape of your DNA. Okay. Franklin concluded that DNA was composed of 2 antiparallel sugar-phosphate backbones with the nitrogenous bases paired in the molecule's interior. All right. The nitrogenous bases are paired in specific combinations. Adenine with thymine, cytosine with guanine. Here we've shown this picture. Here is your sugar phosphate backbone. Okay. Sugar phosphate backbone. Here are your bases, and they are hydrogen bonded together. Okay. Watson and Crick reasoned that there must be additional specificity for pairing, dictated by the structure of the bases. Each base pair forms a different number of hydrogen bonds. Adenine and thymine form 2 bonds. Cytosine and guanine form 3. Okay, here showing your bond between adenine and thymine and guanine and cytosine. Notice that each of these, though they are paired to, adenine to thymine and guanine to cytosine, they are both sets are paired a purine to a pyrimidine. Okay, that is to maintain in the double helix, because remember, it is rotated upon itself. That is to help aid with your forces that hold the double helix together, and it is to maintain an even spacing through your double helix. Okay. Alright, 16.2, we'll go ahead and knock this section out. There's not much to it, I don't believe. This should start to sound familiar as we talk about it. If there's anything that you are having trouble with, please let me know. Alright, so 16.2 is all about the proteins that work together in DNA replication and repair. Relationship between structure and function is manifested in the double helix. Alright, since the two strands of the DNA are complementary, each strand acts as a template for building a new strand in replication. Okay, in DNA replication, the parent molecule unwinds and 2 new daughter strands are built based on base pairing rules. So here's showing your parent strand. It unwinds and separates. Here, your base pairing rules, a new strand is created. It comes in and fills those gaps, and you end up with 2 new strands. Okay? Remember, just follow for DNA A to T, G to C. Alright, DNA replication is semi-conservative. Each of the 2 new daughter molecules will have 1 old strand derived from the parent molecule and 1 newly made strand. So it is semi-conservative because it conserves half of those base pairs from the parent strand in each of the new strands created. Alright, experiments performed by forgive me if I mispronounce this, Massillon and Stahl. Alright, their experiments supported the semi-conservative model of DNA replication. Okay, that experiment is located on page, there we go, 312. Okay, and what this experiment looked at was comparing, let's see, make sure I'm telling you the right thing here, sorry guys. Alright, at California Institute of Technology, Michelson and Stahl cultured E. coli for several generations in a medium containing containing nucleotide precursors labeled with a heavy isotope of nitrogen, N15. Okay, N15 is a heavy isotope. Scientists then transferred the bacteria to a medium with only N14, a lighter isotope. The 2 DNA samples were taken from the flask, one at 20 minutes and one at 40 minutes, after the first and second replications, respectively. Alright? They could distinguish that the DNA of different densities by centrifuging and extracting the DNA from the bacteria. So, this part of the this part of the experiment, the bacteria were cultured in their two new media. Alright, what they found were that DNA sample centrifuged after 20 minutes, okay, after the first replication. Your band is here. DNA samples centrifuged after 40 minutes after second replication. This is less to more dense, okay? The bands in these two centrifuge tubes represent the results of centrifuging two DNA samples from the flask in step 2. One sample after 20 and one after 40 minutes. What they concluded, they were able to compare their results to those predicted by each of the three models that we'll look at in just a moment. You can refer to figure 16.10 for help there. The first replication, the 14N medium, produced a band of hybrid 14 to 15 DNA. This result eliminated the conservative model. The second replication produced both light and hybrid DNA, a result that refuted the dispersive model and supported the semi-conservative model. They therefore concluded that DNA replication is semi-conservative. Okay, let's see if hopefully they have. There we go. Here's showing those conclusions, that it's semi-conservative. Notice in first replication, you had N14 and 15 both present. So that did not support what we were looking for, because they ended up with fully N14 and fully N15 strands. Second replication, same thing. The second replication, we also end up with a semi-conservative. This would be your models here. Okay, and this is what these are our models. Here's your examples. This is what we ended up with. Okay, semi-conservative model we see holds true. Okay, you end up with a hybrid, so you have a 14 and 15 mixed together. And dispersive means that it spreads them throughout all four, and that did not occur in this experiment. Okay? Alright, a closer look at replication. Hopefully this will sound familiar to you guys. If not, make a list of questions. Okay? The copying of DNA is remarkable in its speed and accuracy. More than a dozen enzymes and other proteins participate in this DNA replication. The replication of the DNA molecule begins at special sites called origins of replication, where the two strands are separated. Okay? A eukaryotic chromosome may have hundreds or even thousands of replication origins. Alright, the replication bubble is what they call it, and that's where the double helix opens up, and you have your parental templates, and then what bases are added to complete the strand again are your new daughter strand. The area and direction that that replication moves is based on this replication fork. Alright, the bubbles expand laterally as DNA replication proceeds in both directions. Okay? Eventually, replication bubble fuses and the synthesis of the two daughter strands is complete. So, you have several of these little replication bubbles that pop up along the DNA that is working, and as the replication bubble opens further and that replication fork moves along the strand, unwinding the strand ahead of the bubble, the bubbles will eventually meet up and fuse. Okay? So in eukaryotes, DNA replication begins at many sites along the giant DNA molecule of each chromosome. Alright, and this micrograph is able to show you that. Okay? Elongating a new strand of DNA at a replication fork is catalyzed by enzymes called DNA polymerases, which adds nucleotides to the 3' end of a growing strand. Okay? So, your template strand contains your bases that need matching. Alright. And then your growing strand is your new strand. And it is adding bases to be able to grow that new strand. Okay? How does the antiparallel structure of the DNA helix affect replication? Alright, I want you to ponder that question. That's going to be your one and only for chapter 16 checkpoint question. I want you to give me a good explanation for this question. How does the antiparallel structure of the double helix affect replication? Okay. DNA polymerases add nucleotides only to the free 3' end of a growing strand, along one template strand of DNA, the leading strand. DNA polymerase 3 can synthesize a complementary strand continuously moving toward the replication fork. Alright, to elongate the other new strand of DNA, the lagging strand, DNA polymerase 3 must work in the direction away from the replication fork. The lagging strand, synthesized as a series of segments called Okazaki fragments, which are then joined together by DNA ligase. Alright? Synthesis of leading and lagging strands during replication is shown in figure 16.14. Okay? The same proteins function at both replication forks in a replication bubble. For simplicity, only one fork is shown in this picture. Okay? Only one fork is shown. Go to the left. Okay, let's see. Okay, what's happening here? Your DNA polymerase 3. There's your enzyme there. Only elongates DNA strands in the 5' to 3' direction. Okay, one new strand, the leading strand, can elongate continuously 5' to 3'. So 5' to 3', 5' to 3'. Okay? So one new strand, the leading strand, can elongate continuously 5' to 3' as the replication fork progresses. The other new strand, the lagging strand, must grow in an overall 3' to 5' direction by adding short segments of Okazaki fragments that grow 5' to 3', numbered here in the order they were made. Okay, this is shown here. Okay, all right. So your DNA ligases join the Okazaki fragments by forming a bond between their free ends. This results in a continuous strand. Okay. Priming DNA synthesis. DNA polymerase cannot initiate the synthesis of a polynucleotide, can only add nucleotides to the 3' end. The initial nucleotide strand is an RNA or DNA primer. Only one primer is needed for synthesis of the leading strand, but for synthesis of the lagging strand, each Okazaki fragment must be primed separately. Alright, here is the figure on 16.16 on page 316. Forgive the numbers not quite matching up. This is from the older edition of the textbook. Same information, just slightly different order of the pictures. Alright, what this diagram is showing is synthesis of that lagging strand. The first thing that has to happen, primase joins RNA nucleotides in a primer. Alright, DNA polymerase 3 adds the DNA nucleotides to the primer, forming the Okazaki fragments. Okay. After reaching the next RNA primer to the right, the DNA polymerase 3 detaches. After fragment 2 is primed, DNA polymerase 3 adds DNA nucleotides until it reaches the fragment 1 primer and detaches. Okay? The DNA polymerase 1 replicates the RNA with DNA, adding the 3' end of fragment 2. Alright, the next thing that happens, DNA ligase forms a bond between the newest DNA and the DNA fragment 1. The lagging strand in this region is now complete. Okay, so study that figure there. Use the text around it to help. So let me know if you have any questions on this section, okay? Alright, other proteins that assist in DNA replication. There's a whole chart for you. Helicase, single-strand binding protein, topoisomerase, primase, DNA polymerase 3 and 1, and DNA ligase. This chart tells you a little about the function and how it affects the leading and lagging strands. Okay? This figure is 16.17 in your textbook, and it is showing you a summary of bacterial DNA replication. Alright, this detailed diagram shows one replication fork, but as indicated in the overview, these previous pictures, alright, replication usually occurs simultaneously at 2 forks, one at either end of a replication bubble. Viewing each daughter strand in its entirety in the overview, you can see that half of it is made continuously as the leading strand, while the other half on the other side of the bubble is synthesized in fragments. That's the lagging strand. Okay, so your lagging strand happens in fragments. Okay. Alright, DNA replication machinery is a stationary complex. The various proteins that participate in DNA replication form a single large complex DNA replication machine. DNA replication machine is probably stationary during the replication process. DNA polymerase proofreads newly made DNA, replacing any incorrect nucleotides. In a mismatch repair of DNA, repair of enzymes corrects errors in base pairing. Alright, in nucleotide excision repair, enzymes cut out and replace your damaged stretches of DNA. So excision excises those damaged areas and comes back and synthesizes that little section of DNA so that there are no missing nucleotides. Alright, and then you end up with a new newly repaired section of DNA thanks to DNA ligase sealing up the free end of the DNA so that the strand is complete. Okay? The ends of the eukaryotic chromosomal DNA get shorter with each round of replication. Here's showing you. In our textbook, this figure is 16.19. Okay, if you want to take a look at this, it's showing you the ends of the eukaryotic chromosomal DNA get shorter with each round. And that is because your leading strand and lagging strand, alright, are fragmented by the RNA primer. The primer must be removed and cannot be replaced with DNA because no 3' end is available for the DNA polymerase. So removal of primers and replacement with DNA where a 3' end is available. Alright, second round of replication, you have a new leading and lagging strand, but again, the primer is removed and cannot be replaced because of the direction of replication. So your leading and lagging strands are getting shorter and shorter through the further rounds of replication. Alright? Eukaryotic chromosomal DNA molecules have at their ends nucleotide sequences called telomeres. They postpone the erosion of genes near the ends of DNA molecules. Alright, if the chromosome of germ cells became shorter in every cycle, essential genes would eventually be missing from the gametes they produce. An enzyme called telomerase catalyzes the lengthening of telomeres in germ cells. So, those little red dots are your telomeres. That keeps us from losing essential DNA. Thanks, guys. Make sure you do your checkpoints.